So welcome in another video lecture series. So in this video lecture series we will going to discuss about staphylococcal aureus. Staphylococcal aureus, also known as staph aureus. So staphylococcal aureus, if we break the name or if we try to decode its name, then we will end up with golden cluster of grapes. Okay, so Staphylococcus aureus, if we focus in the name, then we can get that the term coccus. That means it is clear that the staphylococcal aureus is round shape. Yes, is round shape. Okay, and it, as we, after decoding the name of the staphylococcal aureus, we get golden cluster of grapes. Then that means it is clear that it will appear in cluster, right? That is why under the microscope you can see golden cluster of grapes as staphylococcus, right? Because as it is round in shape and they are growing in cluster, that is why they look like grape. Okay, so now after gram staining we get the data that staphylococcal aureus is gram positive. That means what? It will stain purple and it have a thick peptidoglycan cell wall. Okay, so now what is the culture media for staphylococcal aureus? So staphylococcal aureus can be grown in blood agar media. Okay, and in the blood agar media the color of the colony of the staphylococcal aureus is distinctive golden yellow color. Okay, so up to that part we have learned that coccus, that means Staphylococcus aureus, is round shape and they appear in cluster. That is why the name golden cluster of grapes, and they are grown in blood agar media, and in the media the colonies of Staphylococcal aureus appear as a distinctive golden yellow color. Okay, so now they are facultative non-motile anaerobes. So what is the meaning of the facultative anaerobe? Facultative anaerobe means they prefer oxygen free environment, but they can also grow in aerobic environment too. Okay, and they are non-motile, that means they cannot move and they do not form spores. Okay, so up to that part we have learned that staphylococcal aureus, if we decode the name of the staphylococcal aureus, then we end up with the term golden cluster of grapes. That is why, because if you focus the word coccus, that means round shaped. Okay, and as it is appeared in clusters, that is why it looks like golden cluster of grapes. Okay, it is gram positive, it stained purple after gram staining. That means it has a thick peptidoglycan layer in its cell wall. Okay, and it is facultative non-motile and anaerobe and do not have the ability to form spores. Okay, now a important thing that you have to remember. Okay, the first thing is how you can distinguish between Staphylococcus aureus and other type of coccus, let's say Enterococcus, Streptococcus and other type of cocci. Okay, so let me told you that Staphylococcal aureus produces a special type of enzyme called catalase. Okay, these catalase have the special ability to convert hydrogen peroxide into water and oxygen. Okay, so now remember it very carefully that only Staphylococcal aureus have the ability to produce catalase. Okay, other type of cocci do not have this ability. So you can use a small drop of hydrogen peroxide to distinguish between Staphylococcal aureus and other type of cocci, let's say Streptococcus, Enterococcus and other cocci. So what you get as a result, so after putting drops of hydrogen peroxide, you will get foam formation in catalase positive bacteria and nothing will be appeared in catalase negative bacteria. So this is the kind of foam formation that you can get during catalase. Okay, although this is our experiment that we have performed in our university. So here we have used E. coli and Bacillus subtilis as other bacteria, so it is quite difficult for you to understand about what is the difference that you may observe when you use Staphylococcus and other type of Enterococci. Okay, but I think you can understand this clearly that such type of foam will occur for catalase positive bacteria. Okay, so why I have said that for you it is difficult to distinguish with the help of this picture, because here the specimens that we have used for the experiment all are catalase positive: Staphylococcal aureus, E. coli and Bacillus subtilis. So if you use here let's say Streptococcus, then you can get a normal type of solution there. Okay, so that is how the foam formation look like, I have told you here. Okay, so that is what I have written here, that nothing will happen for catalase negative bacteria. Now there is another problem that scientists face, that yes, we can distinguish Staphylococcus from other Enterococcus or other cocci type bacteria with the help of catalase. Yes, that's true, no problem. But distinguishing of Staphylococcal aureus with Staphylococcal epidermidis or Staphylococcal saprophyticus becomes difficult, because they are all catalase positive. That means the condition resembles that I have seen you just, right, that the three specimen you have used and all the three specimen showing foam formation, right? So under such circumstances you cannot use catalase for your experiment, right? So except that you can use another type of enzyme, that is coagulase. Okay, that is another type of enzyme produced by Staphylococcal aureus that you can use to distinguish between Staphylococcal aureus and other type of bacteria. So what is the function of the coagulase? Coagulase have the ability to convert fibrinogen into fibrin. Okay, and fibrin play a important role in clot formation. Okay, so now let's try to think about this, that if you stir Staphylococcal bacteria in a liquid emulsion and then add plasma which contains fibrinogen, okay, you can get two type of situation. What? In case of the Staphylococcal aureus bacteria, you will get clump formation. Okay, and in case of other Staphylococcal bacteria, clump formation will not occur. Okay, because Staphylococcal aureus have the ability to produce coagulase. This coagulase will convert the fibrinogen into sticky fibrin, and that is the reason of the formation of the clump. Okay, and you cannot get this in coagulase negative. Okay, and that's how you can separate Staphylococcal aureus from Staphylococcal epidermidis and Staphylococcal saprophyticus. Okay, so that is the introductory video of Staphylococcal aureus. So Staphylococcal aureus is a cocci, is a round shaped bacteria, and it appears like clusters. Okay, and they grow in blood agar media, right, and they appear golden distinct color or distinct golden yellow color in the blood agar. Okay, so now they are facultative anaerobe, no doubt about this, and they form spores. So these Staphylococcal aureus bacteria have the ability to produce catalase, that helping us to distinguish Staphylococcal aureus from other type of cocci. But when we are willing to separate Staphylococcal aureus from other Staphylococcus, then we have to take coagulase for our experiment. Okay, so this coagulase will facilitate the clump formation in case of Staphylococcal aureus, but clump formation will not occur in other Staphylococcus. So that is the idea for Staphylococcal aureus introductory video. So I hope this video will be helpful to you. Thank you.